Critical Thought TV. Stuart Mason Devart, your host, and today we're speaking with Dr. Randal Koene, neuroengineer and neuroscientist, about substrate-independent minds, whole brain emulation, and mind uploading. And one of the questions I have, Randal, is in terms of the actual structure and function of the brain, what I've seen described as the connectome. I was going to ask you if you could explicate a little bit about the functional and structural components and important issues in substrate-independent minds having to do with the structural and functional connectomes.
Right. Okay, this comes down to the crux of how do you capture the information that's inside, that is being processed inside the brain, or the processes, how they're taking place, and put them somewhere else. That's called a system identification problem. The idea is that you have an unknown piece of machinery, an unknown process that you could describe as a black box. You know that there's input going into it and there's output coming out of it, and you want to know what's going on inside. Now you can describe it either by taking the entire thing, like the entire brain, as a black box and just looking at its behavior and try to describe what's going on. If you do that, then you have a massively complicated system, and you'd have to run it through so many possible input combinations to get any idea of what's going on inside that that's not reasonable. That's why you try to break it up into smaller pieces. That's why you want to take a resolution that's much smaller. And so one that comes to mind, one that seems useful from our perspective, because you know how to measure things when it comes to neurons, is the neuron, so the neuronal level. You want to understand what's going on in each one of those. When you look at it as a system identification problem, once you put your black box around that neuron, and that's the element, then you want to describe the function that that is carrying out. You want to understand, you characterize this particular neuron, what sort of a function is it producing. And then, above that level, you want to understand how they all operate together. How do they interact to produce emergent function? And that's where the connectome comes in, because you simplify your emergent function problem by understanding which neuron is connected with which other neuron and how strongly.
So if I take it correctly, then the whole brain emulation instantiation of substrate-independent minds in this capacity has to do with replicating at the most computationally efficient level of scale in terms of structure and the most reliable measure in terms of function in these two aspects of the connectome. Is that fair?
When you use the word measure, that's really the most important part, because the hard problem of whole brain emulation right now is the data acquisition part. So it's how do you get at all this data from all those neurons. There are 86 billion neurons in the human brain. So you can get a lot about the structure by looking at it. So if you take electron microscope slices of the brain and you stack them all up, you can reproduce the spatial, you can reconstruct the spatial brain. You can get an idea of what's connected with what. But you also need to know what's going on in each one of those elements. That's how you make your system identification problem so easy that you can correct any of the parameters from that spatial model you've created and produce something that works. If you don't do it that way, it is too difficult. It's computationally intractable at the moment.
So that would be analogous, if I'm understanding you, to looking at an integrated circuit and knowing what some of the behaviors are, some of the voltages, the current flows, without having to really worry about quantum tunneling, for example.
Right, because sure, there are these transistors, they use quantum tunneling, but that's really not the crux of the matter. The point there is that you've got things that you define as 1 and 0, different voltages, 0 and 5 volt, for example, or 0 and 3 volt. And you just want to know at every moment, at every clock tick in that system, what are the zeros and what are the ones, and how are they connected? So how can they influence one another? That's what you want to know in an integrated circuit. In a brain, it's a similar system. What really affects everything is the spikes. It's these times when the neurons fire, spike timing. That's what the currency of the brain is really. That is what determines when memories get laid down. So if two neurons spike close together in time, then the connection between them gets strengthened. That's where you put a memory. Right. And the same thing in terms of controlling muscles. So when we control muscles, like when I'm speaking right now, I'm controlling the muscles because the neurons, the motor neurons, are spiking. The spikes of the neurons control the muscles, controls my interaction with the universe around me. So those two things, muscle control and laying down memory, that's why that's the important thing. Yeah.
So would it be fair to say then that the structure implies a range of potential functions, and they're connected obviously, and then the function, the functional connectome then allows you to look at a structural configuration to know what that particular configuration is doing given the spike intervals for that neuronal structure,
yeah, so
more or less.
You really want to reconstruct, or you want to be able to predict the spiking. If you have a system, if you've copied enough information from that system so that you can predict how it would spike in the next moment or when the next spiking would occur, then you've achieved whole brain emulation. And what you need for that is the structure about how things connect, so which functions they can carry out, and the functional characterization within the neuron itself.
Very good. That's very clear. Oh, another question that I have is when I was reading your material about what you refer to as cybercells or micro-neuro interfaces. I found this fascinating, especially the description of one of these devices, which was silicon with a lot of functionality, which I'll leave to you to describe, and encased in a silicon or biocompatible encasing that would almost at least have the dimensions, let's say, of a red blood cell, and would hopefully be flexible in terms of the circuitry to allow the conformation and deformation of a red blood cell so it could get into capillaries and other places, and then perform the kinds of scanning and other interactive functions that we need to capture what we need to capture for whole brain emulation.
Right. Yeah, that's really what it's about. It's trying to solve the one big gap that we have right now in terms of tools. We need something that can do functional characterization at high resolution throughout the entire brain. And you've got many different ways of doing it, and there are alternatives. There are biological ways of approaching this, for example. Right. But the advantage of having this integrated circuit technology, that cyborg cell or micro-neuro interface, is that we really know how to work with integrated circuits. We're experts at building hierarchies and systems with them, communication, getting them to do exactly what we want without too much searching around in the space of possibilities. So we're good at engineering them. And we can make them very small. These days, if you want to put transistors on something that is the size of a red blood cell, you can easily put on there as many transistors as you need for either one of the old Intel CPUs, one of the early ones, or say the guidance system of a cruise missile can fit on that. So there's plenty of room to engineer a useful system.
That's, so then that means, before we move on to the next topic, that we don't necessarily have to or even want to resort to or employ nanostructural devices because of the difficulty in constructing them in a way that they do what we want them to do reliably. Is that a fair statement?
That's a fair statement. Nanotechnology has always been heralded as basically the technology to go to when something becomes really, really hard. And yes, it's like it's the ultimate thing you'd like to have. You'd like to have little nanorobots sitting in your brain next to every neuron and doing whatever you need them to do. But we're just not very good at doing that yet. We're not great at developing nanotechnology, even though there's lots of progress happening there. And we're not so great at building robots either. So having nanorobots is like two big problems putting them together. But this, at the micro level, doing integrated circuitry, that's something we can do today. So that's why the emphasis on that.
If you were to be asked this and you had to give an answer, if you will, which is not quite here, you can defer if you'd like, but what would it take, and do you have a time frame in your mind where you actually expect to see a functional cybercell or micro-neuro interface?
Right. Yeah, I'd be happy to talk about that. Actually, when you look at some of the presentations I've given about this topic, you'll see that it's composed of mostly technologies that have already been developed. People have already put chips inside of cells. People have already built small systems that can run on infrared energy and things like that. So it's a matter of putting things together. To make a prototype that's maybe a little larger than you'd like to have it, and that can maybe not measure everything and is hard to localize where you want to put it, you could do that probably within a year. That's the first prototype experiment could be done that quickly. And then, say, 5 years to get to something that is really usable, where you'd have thousands maybe that you can put inside of a system and start measuring something really useful. It's basically brain-computer interface technology.
And I would imagine, just to ask the last part of this question, that there could be almost a fleet with differentiated functions. There could be one that perhaps performed a lot of the external communication with some type of a computational system outside of the body, but also communicated with and perhaps even directed smaller, more specialized cyber cells.
Yeah.
And so they would have their own, it would almost be like a shadow network.
Yes.
With its
Yeah.
own emergent properties that would, interrogate and communicate and measure the properties of our own biological neural network, communicate that to a more powerful exogenous host, which could then feed back corrective direction to the leader, something like that.
Of the idea. That's why it's so attractive, because this is built on a system where we can easily do that sort of thing. Hierarchical systems, communication, specialization between different things, like we have different components of our computing systems right now. So that's the advantage of using a technology that we're good at, that's a mature technology.
Thank you so much.
Thanks.